Namaste. हमेशा खुश रहो बिटिया. I am Dr. Lily Trivedi, and today I am going to take a lecture on the lab experiments of agriculture B.Sc AG second SEM Fundamentals of Crop Physiology Lab. And the experiment which we are going to take today is measurement of root pressure and rate of transpiration. Now, as you all know, what is root pressure? We all know that Stephen Hales in 1727 observed that the liquid could rise to a height of 21 feet from the cut end of a vine connected to a tube of 1/4 inch diameter. Hence, the root pressure is also described as exudation pressure. Now, how can it be demonstrated? Up The root pressure can be demonstrated experimentally by cutting the stem of a well-watered herbaceous plant. Suppose you have a plant, you can perform this experiment from your house also. There is no complication. You grow a tomato plant and water it nicely, है ना? And then we attach the cut end of the stump. We cut the uh, pot, the stem of the well watered herbaceous plant, and to that end of the stump we will attach a narrow glass tube by the means of rubber tubing. So we pour a little colored water into the tube and its level is marked as A. You can also put a drop of non-drying oil into the tube to prevent the evaporation after few hours. After a few hours of keeping the apparatus, we keep the apparatus in a moist and shady place in the glass tube from the root system, which must be absorbed from the soil by forces present in the root. And what is the forces? That is active water absorption. The rise of water also indicates that the rate of pressure at which the water enters the xylem root. So you have kept a pot and we have kept a tomato plant. We have watered it and cut. Then we cut the tomato plant a few inches above the soil. Now this cut end of the stump to which we attach a glass tube by rubber tubing. We fill some water on the glass and we also put a color water so that we can observe the rise of water. We can also put a drop of non-drying oil to prevent evaporation of water, and after a few hours we see there is a rise in the level of the water. Now this rise in the level of the water is the amount of water the roots have absorbed within the given time. So here you can see the glass tube and the rubber joint and the stump of the plant. And in this glass tube, we are measuring the rise of water. So as you all have seen that the root pressure can be measured by attaching suitable mercury manometer also to the cut of stem. Now the root pressure, it provides the force causing pushing of water into the stem. Now the continuous water flow is maintained in plants because how? Because the leaves transpire water through stomata. And to replace this water, the roots draw water from the soil. And from the roots, the water moves up towards the stem. Now we have a second experiment, that is rate of transpiration. So we have to study the rate of transpiration in leaves, and for this we require a photometer. Now what is a photometer? As from the diagram you can see, it's a U tubing, and on one end it's an airtight seal is maintained and we put a plant cutting. We count the number of leaves in it and the size of the leaf. On this tube at the other end is a calibrated pipe. So this is the basic structure of a photometer. So plant cutting is required. A calibrated pipette is required. Why? To measure the amount of water loss, and the length of clear plastic tubing and airtight seal between the plant and water tubing. So we will place 4 photometers in different environments like high temperature, sunlight, shade, uh, high humidity area, low humidity area, and in different 4 environments we can select according to our choice. Now we will measure the water loss of in every 3 minutes for 30 minutes. For half an hour, for every 3 minutes, we are going to measure how much the level has gone down. We will also calculate the surface area for each cutting of the leaf. Now how we will calculate the surface area? We will lay the leaves on a 1 centimeter grid and trace their outline. Then we will count the number of square centimeters and estimate the partial squares. Suppose if you count a partial square, if it is at least half covered by leaf, we will count it. If it is not covered less than half, we will not count that square. So only the leaf area has to be counted, not the stem or leaf. So from this basis, we come to know the area of the leaf, and accordingly we will calculate how much water loss has taken place. So photometer is a device which measures the rate at which the plant draws up water. Now since plant can draw up water as it loses water by transpiration, so we are able to measure how much water it is absorbing. The rate of transpiration is measured as the amount of water lost. Now the water evaporates through many stomata on the leaf surface, and this rate of transpiration is directly related to surface area. So this is why we are calculating the surface area of each leaf attached to the stem. So to arrive at the rate of transpiration, we must calculate the surface area of the leaf. And most of the stomata are found in the lower surface, and we will determine that surface area. So I hope you have got the basic concept of both the experiments, and we are using calibrated tubing, glass tubing, uh, to measure the amount of water absorbed in the root pressure and amount of water transpired in the rate of transpiration. So thank you very much. I hope you found my session interesting, and if you have any query, please write in the comment box. This digital session was powered by digital version 2.0 of TRIVEDI FAV. Namaste.